5, 6.
Yeah.
So that's, you're just gonna have to look over somebody's shoulder and just as best you can. And then I will scan page 19 and post it on Canvas before I leave, okay? All right, let's get going. We gotta get going. Our bookstore is managed by a company called Colette. And ever since they took it over, we've had nothing but trouble, just like this. The administration has had complaint after complaint after complaint for years. They're not gonna do anything about it. So if you have other complaints about the bookstore, I would tell you to go talk to somebody. But this administration doesn't give a shit about you. So it's a waste of time. I know, because I spent 3 years complaining about this. And it hurts students, they don't care. They make money off of full life. That's why they're not gonna change. Everybody got it? All right, on the bottom of your lab exams is your grade to date. That's on the middle whiteboard. If you have an asterisk, that means you have enough points for a C, but you have to pass a lecture exam, which you get a C. And until you do, it's officially a D. Now let me show you what pages we got, not next week, but the following week. We have lab exam 2 on muscles, which is on page 16, 17, 18. Everybody got those pages? Okay, let me tell you what pages are going to be on lab exam 3. Are you ready? 19, which you don't have. 20, 21, 22, 23, 24, 25, 26, 27, 28. Class starts ramping up. Fortunately, not all those pages are lists. For example, the page you're missing is our list for today. 20 and 21 are pictures that support that. Next week, what we do, the list is 22 and 23, and then there's 2 picture pages. And then the week after that is 26 and 27, and then the picture page. So they're not all lists, but it's still a little bit more than what these do. Okay? Are we ready? Okay, for those that have page 19, again, try to share with a classmate if they don't have it. And you see how there's several sections here we're going to cover? The second one from the bottom says spinal cord cross-section. Do you see that? Spinal cord cross-section? To the left of that, right, page 20 and 21. The pictures on page 20 and 21 is for that section, spinal cord cross-section. Okay? All right. Now we're going to start out with the neuron. And the model that's on the screen is the model that's on the front desk. Blue arrows are on the dendrites. In the red line is the cell body, or soma. And then the green arrow is pointing to the axon. And those are the 3 main regions listed on the top of page 19. You guys remember the classification of neurons? Unipolar, bipolar, multipolar? Which one is this one? It's multipolar. Multiple dendrites, 1 axon. Each dendrite is a pole, multipolar. Okay, we could open up the cell body, and here we see the cell body in red. In the middle is the nucleus, it's the purple arrow. And all these orange arrows are on these little packets of rough endoplasmic reticulum and ribosomes called chromatophilic substances, full name Nisselbody. Be very careful with Nisselbody. It's only one vowel, 2 syllables.
Where is it? Oh, Nissel?
Each one of these is bluish gray in color, which is what gives the cell body a bluish gray color. So when you get a whole bunch of cell bodies together, that's what makes gray matter gray, is the cell bodies. And it's from these little packets in the orange arrow. Everybody got it?
Mm-hmm.
Any questions? All right, there's the axon again, but it's only this brown structure that's in the middle. That's the part of the neuron. This model does not have 1 cell, it has 3 cells. Because the blue arrow is pointing to that different cell called the neural lemmacyte. Now the light blue arrow is just pointing to the nucleus of the neural lemmacyte. That's why this model has 3 nuclei on it. There are 3 different cells. The outermost layer of the cell is called the neurolemma. The neurolemma is the yellow arrow. But remember, this neurolemmacyte has wrapped itself around the axon over and over and over again. And the thickness of that wrapping is called the myelin sheath, and that's in red. So the outermost phospholipid bilayer of that myelin sheath is the neurolemma.
Is that also the other white stuff that's not has an arrow to it?
No, that is,
Okay.
I think the key says it's endodermal. That's why it's like collagen fibers.
So it's pointing to that pink, that thin pink layer on
Yes.
top. Okay.
Right where the tip of the yellow arrow is. Everybody got these? Okay, this picture is the cross section here. Everybody see that? That's what you're looking at on the screen, right? Let's get right here at the end. Axon is in green, neurolemma is in yellow, red is myelin sheath. But then you see these brown dots. They're actually brown lines, but since this is in cross section, you're only seeing the ends of those lines. That's why they look like brown dots. And those are neural fibrils. That's the purple arrows, neural fibrils.
Not the notes, right? Just neural fibrils. Okay.
All right, so here's the axon, which is part of the neuron. The yellow arrows are on the nuclei of the 2 neural lemmacytes. Just write neural lemmacytes, you don't have to say nucleus. But they are different cells, that's why they have their own nucleus. The gap between adjacent neural lemmacytes is called the neural fibril node, or node of Ranvier. That's the orange arrow. Okay, everybody got these?
Is the node where they come together, or is it the
It's
pink
the gap
part that
between them.
gaps them? Okay.
Remember how I drew it on the whiteboard on Tuesday? There was a gap between the myelin.
Those are the light blue one?
What's that?
The light blue one?
The light blue ones are the 2 neural lemmacytes. They're specifically on the nuclei, but you don't have to say nuclei because that's not on page 19. It's just showing you these are the cells, the neural lemmacytes, and the gap between them is the neural fibril node. Okay? Now here you can see some of the brown lines. And in fact, can you see the brown lines here? These are protein filaments that give a multipolar neuron its shape. those brown lines are the neural fibrils. And so you can see some of the ends right here where it's cut in cross section. So they're either lines going longitudinally, or in the cross section they're brown dots, because they're the ends of the line. All right, now let's talk about anatomical types of neurons, and the second section is about how many poles come out of the cell body. This guy right here at 3 o'clock from the cell body to the right, there's one pole. This is a unipolar neuron. And you can see this thing that's on the chair right here. Okay, everybody see the one pole coming off to the right? And I said that bipolar neurons are in the retina and the inner ear. They're pretty rare. So in the bottom left of that chart on the chair is this, which is the retina. And you can see this cell right here has 2 poles coming out of the cell body. Can you see that? One at 12 o'clock, one at 6 o'clock. That's a bipolar neuron. Retina and inner ear is where we find them. And then both of those guys are multipolar neurons, just like this guy. Multiple dendrites, 1 axon. That would be what they look like, anatomical classifications of neurons. Now this is the functional classification of neurons. Which way are the action potentials flowing? Here you have to identify the CNS, which is down here. This is the CNS. So if the action potentials in green there are going into the CNS, that's a sensory neuron. And in fact, if you look at unipolar, up on top, above there, sensory neurons are unipolar. Everybody see that? Where it says unipolar, blah, 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 e.g. sensory neurons. Sensory neurons, like this, are unipolar. That'll come into play later when we talk about the spinal cord. If action potentials are coming out of the CNS, like this red one, that is a motor neuron. And you need to draw your own arrow, but this blue neuron, which is entirely in the CNS, this yellow box, that is an interneuron. Interneurons are entirely in the CNS. Okay, everybody got the functional classifications? All right. Next section is neuroglial cells. What's listed here are the 4, we covered this in lecture today, the 4 that are the CNS neuroglial cells. How many are you responsible for in this class, total neuroglial cells?
4.
5.
Oh.
4 in the CNS, 1 in the PNS. Which one is the one in the PNS? Because we already covered it in lab. Like 10 minutes ago. So it's obviously above where we are now on page 19. Like on the top right. Third thing listed under axon. Neurolemmacyte, there we go. Boy.
Oh my god.
Was that enough hints? So there's the 5th one. So the 5th one's in the PNS. These 4 are in the CNS. Here's how you're going to take note of visual recognition. First of all, the myelinating cell, well, let's skip that. Let's go to the astrocytes. The astrocytes form the blood-brain barrier. So they're going to have these foot processes on a blood vessel, capillary. If you know that astrocytes, blood-brain barrier, you know this has got to be it because it's on a capillary. Everybody see that? Again, in reality, if this was a realistic picture, you would be all green, you would see no red because it's completely covered with the foot plates of the astrocytes. If you see myelination with neural fibril nodes and this cell is branched, that's got to be an oligodendrocyte. It's the myelinating cell in the CNS, but you're looking for neural fibril nodes. We covered this in lecture today, didn't we? Remember that ependymal cells are all about CSF. So if you see a little shoreline here with fluid, and the cells lining the fluid, those have to be ependymal cells because this is CSF, cerebrospinal fluid. It has cilia on the apical end of the cells, that's what's circulating the CSF. And then, by the process of elimination, if you don't see it on a capillary, if you don't see neural fibril nodes, if you don't see it bordering fluid, it's got to be microglia. That's how you visually recognize it. If it's on a blood vessel, got to be an astrocyte. If you see neural fibril nodes, got to be oligodendrocyte. If you see it on the shore of fluid, got to be ependymal cells. If you see none of that, the fourth one has to be microglia.
On the test, will you be showing us these pictures? Or
No. But it'll be obvious if one of the cells is on a blood vessel. It'll be obvious the neural fibril nodes. It'll be obvious if it's on the shoreline of fluid. Doesn't matter which drawing, which cartoon I use. Okay? Any questions? Okay. Now we're going to get to the connective tissue. This is, I think, on the bottom of this card. This is an eighth grade art project. What you want to write next to this line is this is a cross-section of a nerve trunk. Cross-section of a nerve trunk. This nerve trunk has 4 fascicles. Can you see the 4 groupings?
Mm-hmm.
So, around the entire nerve trunk, this blue stuff, the orange arrow is pointing at, has to be epineurium. This beige circle that's surrounding a fascicle, the red arrow is pointing up, that has to be perineurium. Then this gray stuff, the blue arrow is pointing at, is separating individual axons, and that's endoneurium. This is just a close-up. Same color arrows. Epi, peri, endo. This is a old telephone cable for landlines, which are pretty rare now. But it's showing you the reason why you have to have these neuriums. This gray outer covering is like epineurium. Then you got this blue thing that has groups of individual telephone cables in it. This blue stuff is perineurium. And then the individual wires have this rubber on it. That's endoneurium. And you have that so your neighbor can't listen to your phone calls. You have that so action potentials stay where they belong and don't go to other fibers. Everybody got it? Mm-hmm. All right. That was connective tissue in the PNS. Let's turn back to the very beginning. The page in front of histology, page 4, look at that. There's might, since that wasn't on the last page, you might have missed this. Page 4. Go to the bottom of page 4. The anterior cavity had the mesothelium, you remember that? Pleura, pericardium, peritoneum. That's anterior cavity. Now go above it. In the posterior cavity, we have meninges. Everybody see that? That's where meninges belongs. So you have this mesothelium, pleura, pericardium, peritoneum in the anterior cavity. You have meninges in the posterior cavity. Now go back to page 19. And that's what we're going to talk about. The meninges covering the spinal cord. It's a little bit different on the brain. This model here, there's two of them on the middle shelf of the cart. Both of them are going to be on lab exam 3. This is worth 4 points, this model right here. All right. The green arrow is the outermost layer of meninges called a dura mater. Check the spelling, it's mater, 1 T. By the way, it is continuous with epineurium. Epineurium just runs in the dura mater, and you can't tell where one ends and the other begins. It is continuous. So look here. See dura mater, dura mater, dura mater, dura mater. Oh, that's epineurium. You can't tell where one ends and the other begins. Everybody got it?
Mm-hmm.
Now look at the number 11 here. Everybody see that?
Mm-hmm.
Do you see the black line going through it?
Uh-huh.
That's the line between dura mater and arachnoid, which is the middle meningeal layer. That space in between those two layers is very tiny. But what's actually stuck on the spinal cord with the purple arrows is pia mater. Actually stuck on the CNS, pia mater. Now the distance between the pia mater and the arachnoid is rather thick, and that's entirely filled with cerebrospinal fluid so that your CNS is protected in a water bag. Okay. You know when people have spinal cord injuries and they lose their use of their legs, just think how much that would happen if we didn't have this water bag. It's really highly protected for trauma. But since this is cerebrospinal fluid, what are neuroglial cells lining the subarachnoid space? Ependymal cells. It's circulating the cerebrospinal fluid. So this is all lined with ependymal cells, but this is all cerebrospinal fluid protecting the spinal cord. What does arachnoid mean? Spider-like. Separating these two layers are these web-like extensions, and they saw it and they looked, oh, this is like spider webs. That's why they named the middle layer arachnoid. Does that make sense? Okay. Now we're down to the section where you're going to be going to page 20 and 21.
Oh, thank God. That's so good.
Let's see here. Here's part of the spinal cord. If you look at this model here, whenever I'm going to show you a model on this on Lab Exam 3, this is the spinous process. Do you guys know what section of the vertebral column this model comes from?
Uh-huh. Thoracic?
Cervical.
Oh.
Why do you say cervical?
little red circles?
Yeah, this is the transverse foramen right there, that these vessels going through, so this is cervical. And if I were to hold it up on my body where this is taken from, it'd be like this. Everybody see that?
Mm-hmm.
Spinous process is posterior, correct? Next to this slide, maybe write posterior on top. Because on any of the models for lab exam 3, I will put the posterior on top. Posterior is on top. Okay. Yeah, that's scary looking. All right. Okay, if you got the spinal cord here and you go lateral from that, you got posteriors on top, anteriors on bottom. Right next to the spinal cord are roots. The one place where all of it comes together is technically the spinal nerve. You go lateral to that, you got rami. All right, everybody got it? Okay, so the red arrow with the capital A is where all of it is together. That's the only place I can pin that you can call spinal nerve. Okay.
Can you repeat that one more time?
Red arrow, capital A. The only place where all of it is together is the spinal nerve.
Spinal nerve.
But medial to that spinal nerve, in between the spinal nerve and the spinal cord, are roots. Posterior is always on top, so blue is the posterior root, and orange is the anterior root. And what you need to know is that the anterior root is a one-way street of motor. Posterior root is a one-way street of sensory. What kind of neuron is sensory neurons? Look above on page 19. What is it? What kind of anatomical neuron are sensory neurons? Unipolar. And that's why in light blue you have the posterior root ganglion, because that's where the unipolar cell body is. Do you see where I drew it? You don't need one on the anterior side because there's no cell bodies there. Does this help that I'm using the same colors as these guys?
Mm-hmm.
Any questions?
So the That's just why it's fatter.
Okay, the gray butterfly. Now you have to know different parts of the gray butterfly. And this is shown on top of page 21, even though the arrows are kind of messed up. There are 3 gray horns. There's the posterior gray horn, that's the top green arrow. There's a lateral gray horn, that's the middle green arrow. And there's the anterior gray horn, that's the bottom green arrow. Remember, the gray horns are collections of cell bodies, right? Because isn't that what makes gray matter gray? Remember in the histology quiz, it's the whole gray butterfly, you just had to know it as gray matter. Now you have to identify posterior gray horn, lateral gray horn, anterior gray horn. The middle of the gray butterfly is called the gray commissure, and that's the dark orange arrow. But in the middle of the gray commissure is the central canal. And I've kind of drawn it right here. Again, I think all of this is on pictures on page 21. Did everybody get your lab exams? Everybody picked up their lab exam? Everybody got their grades? I can erase that. The reason why there is a central canal, and I'm just telling you, when I took comparative vertebrate embryology, it made everything make sense. Not just how things look, but how things function when you watch things develop embryonically. Well, your entire nervous system, all of it, 100%, started out as a single hollow tube. The whole thing. The central canal is the remnant of the hollow neural tube. That's the hollow space in the tube. Everybody got it? Next week we're going to look at the ventricles and aqueducts of the brain. Same thing. It is the remnant of the hollow space in the hollow neural tube. Okay? These names are going to be more relevant next week on the brain. But we do have these 2 examples on the spinal cord. Deep cracks are called fissures. A shallow crack is called a sulcus. And this is for next week. We don't have any this week. But if you got a ridge or a bulge, it's called a gyrus. But the bottom 2 words are going to be applicable. Okay, the white matter also runs in funiculi, and there's 3 of them, and it parallels the gray horns. So up on top is posterior funiculus, middle is the lateral funiculus, and the one on the bottom is the anterior funiculus. Just remember, posterior's up on top, anterior's on the bottom. Makes this a lot easier. Now, if you look, this model doesn't show it really well, but look on page 21. On the top picture on page 21, you can see on the anterior side, the medium crack is deep, and on the posterior side, the medium crack is shallow. Everybody got it? So now if you're going to follow this, this is the anterior median fissure, and the one up at 12 o'clock is the posterior median sulcus. Only the one on the anterior is deep. Everybody got that? Everyone here know what PMS is? Well, your spinal cord has it. And it's posterior median sulcus, and now you won't get it mixed up. Everybody got all these? All right. Now if we go lateral to the spinal nerve, now we got rami. And 21 is the posterior ramus, 20 is the anterior ramus. But the thing about this manufacturer, it has a couple errors. Number one, it made this one almost as big as this one, and in real life, it's more like that. Posterior ramus is this little twig, and the vast majority of the spinal nerve goes off to the anterior ramus. In fact, when the cadaver dissectors are doing nervous system extractions, oftentimes we'll call it a spinal nerve. It's actually the anterior ramus. Everybody got it? Because it's like most of it anyway. Just like how I drawn it here. Can you see that?
Mhm.
This is anterior ramus. Doesn't it kind of look like the spinal nerve? The only thing is it's after the split off of the posterior ramus, which is tiny. Okay? Any questions? All right, for these other red arrows, I need to go to this model first. And this is the model for the very bottom of page 19. When you guys are studying for this in Open Lab, do not pick this thing up like this. Lie it flat on the bench. Because of this brain, it's really top heavy. And it goes clunk. You got that. You got people getting injured. Just lay this thing down flat because it's really heavy and it's top heavy. Everybody got it? Now look on the right side of this model. Very carefully. Look on the right side of this model. It's not on the left side, it's on the right side. You see what looks like to be a string of pearls. Can you see that? The string is yellow, the pearls are kind of a pink.
Mm-hmm.
You see that back there? String of pearls. The string is yellow, the pearls are kind of pink. Everybody got that? This is the sympathetic trunk with ganglia. They're not the same thing. The trunk is the string, the yellow. The ganglia are the pink pearls. Everybody got it? If you have that down, now you can understand this. See the circumference of this bulge?
Mm-hmm.
This is a pearl. This red arrow is on a sympathetic ganglion. Singular for ganglia. This arrow, sympathetic ganglion. Up here, notice how this circumference is a lot smaller. Everybody see that? This is the string. Up at the very top, there's no arrow on it, but this is the sympathetic trunk. Sympathetic trunk, the string. Sympathetic ganglion, the pearl. Everybody see that? Well, the other mistake this manufacturer made is it just made one connection here. There's actually two, like how I've drawn it here, like how it is on page 20. You can see that on page 20. There's actually two that connects the anterior ramus to the sympathetic trunk and ganglia. So you can call this communicating ramus. But there's actually two of them. It connects the sympathetic trunk and ganglia to the anterior ramus. And 22, it's connecting the sympathetic trunk and ganglius to the anterior ramus, number 20. Everybody got that? Any questions?
No, it says here communicating Rami. Should that,
Rami is plural.
is that how we should say it on there? Because you can't.
You could say it either way, especially when the model has an error.
Okay.
Because it just has one, it's
Right.
supposed to be two.
Okay.
And ganglion is.
Singular for ganglia. Ganglia is plural, ganglion over there on the whiteboard is singular.
Okay.
Any other questions? Okay, I think we're done with this discuss section. This is on the cart, I think, on the bottom. It doesn't have rami. It only goes as far as the spinal nerve here in red, but this is the same color arrows that I had on the previous slide. Posterior root, anterior root. Posterior root ganglion. Posterior is up on top. So here's the gray horns, boom, boom, and boom. With the great commissure and the central canal in the middle of the great commissure. The three funiculi, posterior, lateral, anterior. The deep anterior median fissure. And your spinal cord has PMS. Anybody need any of these things done? Same color arrows as the previous slides. I can go on? All right, now let's talk about this guy here. That's on the bottom, and the numbers next to those structures match the numbers on the key. Everybody see that on the bottom? For those of you who have page 19. There's numbers on all those things, matches the numbers on the key. All right, the word we use for nerve network is plexus. There are 4, but in reality the bottom 2 are the same network. All right, so this is the spinal cord. Here you can see the spinal cord has the anterior median fissure. This is a deep crack. And if you look at this model very carefully, it's not just the spinal cord. Your brainstem has an anterior median fissure. It goes all the way up the brainstem. This is not spinal cord here. This is brainstem. Everybody got it? And you can see the crack all the way, going all the way up the brainstem. All right, the small network in the neck is the cervical plexus. That's the red arrow. The large plexus going into the arm is the brachial plexus. Everyone knows brachium, right? Biceps brachii. Anybody remember that?
Mm-hmm.
This is the close-up of the plexuses there again. And again, on this model's right side, you only put it on the right side, is the sympathetic trunk. Yellow arrow, doing the yellow things. Sympathetic ganglia, blue arrows. Sympathetic trunk, yellow arrows. Sympathetic ganglia, blue arrows. They're not the same thing. I'm sure you know that. From what section of the vertebral column is this picture taken from? Thoracic. Because you got the ribs. Thoracic is the only spot that has ribs, right?
Mm-hmm.
That's why there's rib facets on thoracic vertebra. It looks like you can put that thing on the grill. And this is a model that's up on top of the cart. It shows that the sympathetic trunk and ganglia are just on the other side of the vertebral bodies. Can you see that?
Mm-hmm.
The spinal cord is in the vertebral foramen, and the sympathetic trunk and ganglia are really close by, just on the other side of the vertebral bodies. But this is the string, sympathetic trunk. These are the pearls, sympathetic ganglia. Okay. Now when you get to the lumbar vertebrae, that vertebral foramen is really big, isn't it? The whole thing is big, lumbar vertebrae, correct? I mean, that was just the last time you just got back. Right?
Mm-hmm.
Here's the thing that might surprise you. Past L1, there is no more solid spinal cord. Look at this. This is T12 because that's the last floating rib, correct? L1 is right here. That is where the solid spinal cord ends at L1. It's called the conus. Looks like a little cone. But that's it. Below that, there's no solid spinal cord. It's all anterior rami. And when the anatomist looked at all those anterior rami, and we have done that when we've done nervous system extractions, it looks like a horse's tail. Doesn't that look like a horse's tail?
Mm-hmm.
And that's what the anatomist named it, except in Latin, which is cauda equina.
Oh. Why not call it horse's tail? Connie, where's it in here?
Cauda equina in orange. And the bottom two plexuses here that the model separates out as lumbar plexus and in purple sacral plexus.
Oh, cauda equina. Oh gosh, I can't even.
But look at this network. It's all intermingled. It's all one network. Can you see that? So in lecture, I covered this as the lumbosacral plexus, as one large plexus, because it practically is. And if you want to talk about sciatica, I will give you evidence that it is actually one network, one plexus. So this whole thing here is cauda equina in orange, lumbar plexus, sacral plexus in purple. Coming off the sacral plexus is the largest nerve trunk in our body, and that's the yellow arrow, sciatic nerve. The sciatic nerve is there first, then the ilium forms around it. What does that leave in the ilium?
Greater sciatic.
Greater sciatic notch.
Does it kind of branch up right there, or is it just that one big eye?
It's just this one bit here. This is the only place we can identify it as sciatic nerve.
Oh, okay.
But it's getting sources from the sacral plexus as well as the lumbar plexus. Do you know anybody that has sciatic nerve issues? Sciatic? They usually have weakness in their leg muscles, or they get a tingling feeling down there, way down there in calf, maybe even going down to the foot. The problem is not in the leg. The problem is here, in the lumbar intervertebral discs, in between the lumbar vertebrae. Your vertebra, and the reason why the lumbar is getting bigger and bigger and bigger as you go down, is these vertebrae have to withstand all the cumulative weight above it. So that's why, your cervical vertebrae are not that big. You just got to support your head. As you get into thoracic, they're not that big until you get down in L8 down to 12. I mean, T8 down to T12. Then they're getting bigger and bigger and bigger. T12, it just looks like a lumbar vertebrae, except it's got rib facets. Then L1 is big, L2 is big, L3 is even bigger, because there's cumulative weight above it. Everybody got it?
Mm-hmm.
Over time, these symphyses, fibrocartilage discs, start to compress. And when they compress, they start squeezing on these guys. You follow me?
Mm-hmm.
And that is where the problem of sciatica is. It's not in the leg. It's pinching nerves in inner vertebral discs in the lumbar region. You follow me? Now, your textbook has to pick a plexus where this comes from. And so it actually has to pick it off the sacral plexus. But the sacrum is fused vertebra. You cannot compress disc in the sacral region. You can only compress disc in the lumbar region, because the sacrum is fused, correct?
Mm-hmm.
And that's why it is one plexus, the lumbosacral plexus. That's where the sciatic nerves comes from, and from the lumbar spinal nerves, those are the ones that get pinched that cause sciatica. That make sense?
Yeah.
Well, the model has split it up between lumbar and sacral, so I do this for lab, but when I cover it in lecture, it's lumbosacral plexus. Clear?
Mm-hmm.
Okay. Going right down the middle of the caudiplyna is this thread that doesn't have any axons in it. That's all collagen. And that midline thread is called the filum terminale. The red arrows. It comes off the spinal cord, fuses with the dura mater right here, and it becomes the coccygeal ligament that inserts on the coccyx, and it anchors the inferior CNS. But we don't have coccygeal ligament on page 19, so if I pin that, that's just call it filum terminale. You can see it inserting on the coccyx there, can't you?
Mm-hmm.
Any questions?
Filament terminale. I can't spell it.
Filum terminale.
Oh, okay. Filum, not filament. Oh God. Oh.
Okay, let's come back at 5 after 2 o'clock. We're gonna bring out the cadaver. You have to have all your electronic things put away. Cannot be any pictures or anything that can take a picture out when the cadaver's out. I'm gonna have to move a couple tables around, 2 cadaver dissectors.